So I want to harken back to Peyer's patches and M cells. So we talked briefly when we talked about the adaptive immune system about this lymphoid organ called Peyer's patches. So Peyer's patches surround the intestinal lining. They surround our intestines, and they're filled with lymph, and they're filled with white blood cells because that's what lymph is. So each Peyer's patch is small, but collectively Peyer's patches are the largest lymphatic organ of your body. 80% of your white blood cells exist in their Peyer's patches at any one time. And their role is to preemptively develop immunity against the microbes that reside in your gut. So atop each Peyer's patch sits an M cell. The M cell's job is not to absorb nutrients like the intestinal epithelial cells, but rather to take in antigens from the microbiome, from your gut, and present them to the white blood cells beneath, like B cells and T cells. So this way, you can activate armies of B and T cells preemptively. These microbes have not infiltrated your protective barriers. They're sitting in your gut where they're not hurting you, and they're digesting your food and producing vitamins for you like they're supposed to. But any one of those microbes could cause problems to your health if they got into your body. If they got past this intestinal epithelial cell barrier, they could cause infection. So what your M cells do is it takes in antigens, exposes those antigens to your memory, to your the cells of your adaptive immune system, allows your adaptive immune cells to generate armies that are sitting there laying in wait, ready in case any of these microbes breach your intestinal epithelial barrier and get into the wrong place, so that you have immunity to your intestinal microbiome. So I want to remind you that M cells are specialized to endocytose these microbes, and that's different from M proteins. I have students confuse M cells with M proteins all the time. M cells are cells in your intestine. They're you. M proteins are virulence factors on Staphylococcus species. Okay? They are two completely separate things. An M cell is one of your cells that helps you generate immunity to the microbes that live in and on your body. M proteins are proteins that belong to Staphylococcus species, and they help infect your body. Close-up of those M cells, and we looked at this slide before in the adaptive immunity lecture. And so again, you can see that these are the intestinal epithelial cells designed to take in nutrients. These have the TLR9s that allow for that allow for tolerance to microbes on this side, but intolerance to microbes on this side. And then you have these M cells, whose job it is to endocytose material from the gut and expose that to the Peyer's patches beneath, where all the white blood cells are, so that you can generate immunity preemptively to the microbes in your gut. So antibodies are then formed in the lymph tissue around the intestines and sent to the mucosal lining of the cell wall to fight pathogens before they can even breach a barrier. Okay, so you have this immunity for, in case any of the microbes in your gut breach this barrier into your body, but you are also controlling the growth of these microbes while they're in your gut. So these intestinal epithelial cells have those tight junctions to prevent infection, but there's also a mucus layer that's not shown here. And that mucus layer has a bunch of antibodies that are sent out and that just sit there in the mucus to prevent microbes from penetrating and to help prevent overgrowth of the microbes in your gut. Now, Peyer's patches obviously have this really essential role in helping you maintain a healthy balance between, a healthy boundary between you and your microbiome. And yet, Peyer's patches are much reduced or even totally absent in germ-free mice. Germ-free mice are mice that are raised in sterile housing. And this could partially explain the importance of microbes in developing our immune system. Okay. So I'm going to, in terms of immune system development, I want to go back real quick to this discussion of the TLR9s. So these TLR9s that have different responses to microbes depending on what side of the intestinal epithelial cells you find them on, they have to be programmed. So why would a TLR9 inhibit the NF-kappa B pathway here and initiate it here? We don't fully understand how that programming happens, but we know that it can get mixed up. So we know that people who have irritable bowel diseases often have misfunctioning TLR9s, okay? And that might contribute to their chronic bowel inflammation. So we don't know exactly how these TLR9s are programmed, but it seems like that programming happens in response to exposure to microbes early in life. So we have microbial exposure programming these TLR9s that allow us to have tolerance for our gut microbiome, and we have these Peyer's patches that are developed in response to microbial exposure early in life as well. All right. Additionally, microbial-associated molecular patterns such as peptidoglycan and perhaps other antigens can travel through the bloodstream from the intestine to bone marrow and stimulate B cell production. So in other words, B cells are produced in response to exposure to microbes. This means that when the microbial community and the number of microbes is altered, it changes how many immune cells are even produced. So germ-free mice produce fewer immune cells than mice that are exposed on a regular basis to microbes. And this is another way that microbes train your immune system. So I want to remind you here that T and B cells begin their development in bone marrow. B cells complete their development there, while T cells complete their development in the thymus. It now becomes relevant to tell you that there is a type of B cell called a regulatory B cell. These B cells secrete a cytokine called interleukin 10. Interleukins we've talked about before. There are many different kinds of interleukins, but interleukin 10 is an anti-inflammatory. Thus, the role of regulatory B cells is to regulate immune response so it does not go completely nuts. Remember that an overreactive immune response can be damaging. Too much inflammation and too much fever can be damaging. Gut microbes tend to promote regulatory B cell production, and studies have shown that the use of antibiotics and also sterile housing conditions for mice reduce regulatory B cell production, and this can contribute to mouse arthritis, and it seems to be involved in the development of allergies and asthma in these mice. So regulatory B cells and interleukin 10 are probably implicated in all conditions described by overreactive inflammation responses that have been correlated with dysbiosis in the gut. So just to quickly recap what we just talked about, we have the TLR9s, which give us tolerance for our gut microbes. These are programmed by microbial exposure. That's one way that microbes program your immune response. We have Peyer's patches that give you preemptive immunity to your microbiome in case any of them happen to invade you, and it also helps you control microbial growth in your gut. These only develop in response to microbial exposure. So that's another way your microbiome trains your immune system. Antigens from microbes in your gut stimulate B cell production in general, but especially stimulate regulatory B cell production, which dampens your immune response so your immune response doesn't go haywire. This is yet a third way that your microbiome trains your immune system. Okay. The gut microbiome is not only implicated in the development of immunity, it is also implicated in obesity and nutrition. Transferring the gut microbes from obese mice or obese people into germ-free mice causes the germ-free mice to become obese, which does not happen when microbes from lean mice or lean people are transferred to germ-free mice. So this tells you that gut microbes affect how we digest our food and how we store fat. This is also related to early antibiotic use. Mice given antibiotics very early in life are more prone to obesity. It's not 100%, remember, it's just an increased risk relative to those who did not receive antibiotics. Similarly, we find that antibiotic use on very young mice increases their risk of allergies and asthma as adults. The younger the mouse when antibiotics are administered, the higher their risk of developing these conditions later in life. What is weird about this is that the younger the mouse, the faster their microbiome seems to recover after antibiotic use. So how can this be, that the microbiome recovers quickly with young mice, but they are still at a higher risk of developing these metabolic, immune, and inflammation disorders after antibiotic use? Well, more research is required, but we may have just explained why in the previous slides. Even though the microbiome recovers when the mice are very young, while they're young, their immune systems are developing and in the process of being set in stone. They're being programmed. They are growing their Peyer's patches, the size of which might never reach full potential if not stimulated by microbes. They are programming how their TLR9s respond to specific microbe-associated molecular patterns encountered in the gut. So if the gut microbes are removed at these early programming stages, the effects could be long-lasting. Similarly, these mice are setting up how the production of regulatory B cells responds to the environment. When the mouse's body senses that there are no microbes to fight and no inflammation to regulate, the pathways which regulate inflammation seem not to be properly built. And so, though it takes an adult microbiome much longer to recover after antibiotic use, even though it takes an adult microbiome much longer to recover after antibiotic use, because all of those immune system components are already programmed and already set in place, already set in stone, there are less likely to be long-term effects. Whereas the infant microbiome is rapidly changing, and so it bounces back very quickly, but the interruption in immune system programming and immune system development might not always be recoverable. Okay. So there is a lot of research out there on gut microbiomes with mice. It's much harder to do these kinds of controlled studies in humans. However, what we're looking at in terms of epidemiological studies, when we track, antibiotic use at different ages, tracking what happens later in life, we're finding that the human research matches with what we find in the mouse research. And so that gives a some fairly robust evidence that what we're seeing in mice is also happening in humans. Now, I want to talk a little bit about, we've talked about how the human microbiome affects immune system development, but I also want to talk about specifically critical illness. So this is not just about early immune system programming. This is also about later in life loss of the microbiome when we're talking about antibiotic treatment. So it used to be thought that antibiotics were this miracle cure. You could destroy any pathogen that could once easily, kill you and make it completely harmless to humans through antibiotics. There are microbes, like staph, that used to take limbs, take lives all the time. We don't even think about it anymore because of antibiotics, except now that there are antibiotic-resistant strains. So for a while we thought, antibiotics are the miracle cure, they can only do good and there are no side effects. But now we're finding that's not exactly true. So SIRS describes an overreactive inflammatory response that can lead to multiple organ system failure. It stands for Systemic Inflammatory Response Syndrome. Okay. Inflammation can increase permeability of the intestinal epithelial cells by disrupting tight junctions. So when we talked about inflammation, healthy inflammation, when we talked about the innate immune system, I told you that certain phagocytes can, inflammation makes the tight junctions between blood vessel cells more permeable so that certain phagocytes can leave the bloodstream and go to infected tissue in order to get rid of a disease, an infection. So inflammation does the same thing in your gut. It loosens the tight junctions and it makes those intestinal epithelial cells more permeable. And in the gut, that's kind of bad because you have so many microbes there. Okay. So when you have chronic inflammation in the gut, it makes a patient more at higher risk of death due to sepsis. Now, sepsis is when you have microbes replicating in your bloodstream, which is really, really bad. Instead of a local infection where microbes are just in one part of your body, they're replicating throughout your bloodstream all over your body. Okay? So the problem, because you have so many microbes in your gut, if you have chronic inflammation in your gut, those microbes then are at higher risk of being able to infiltrate the intestinal epithelial barrier because inflammation loosens those tight junctions. Microbes can slip through then, and then they're in your body, and then you have a higher risk of infection throughout the body in sepsis. Studies have shown that people with a greater diversity of microbes in their gut are significantly less prone to death from sepsis, likely due to the protective effect of our good microbes, which prevent pathogens from establishing by competing and fighting with them. Several standard ICU treatments have been shown to alter the gut microbiome, usually simply by altering the environmental conditions of the intestine. So vasopressors can cause local hypoxia or low oxygen conditions, and they also lower the pH. Acid suppressive therapies alter the pH of the intestine. Highly processed enteral nutrition or parental nutrition can cause a nutrient deficient milieu in the bowel. So basically, if you're changing the nutrients in the gut, if you're changing the pH of the gut, if you're changing the oxygen concentrations in the gut, all of those kinds of things are going to alter the gut microbiome because every microbe has optimal conditions in which it grows. And if you change those conditions, it's going to change what microbes can grow there. Long-term opiate use is associated with bacterial overgrowth. And lastly, of course, antibiotic use causes depletion of the gut flora and potential selection of resistant organisms. So if you have, again, these are still ideas that we're exploring. We're trying to figure out the mechanisms. But the more diverse your microbiome, the less prone you are to SIRS, which means the less risk you are of sepsis, of death from sepsis when in the ICU. And so it's not that you can always avoid antibiotic use because, if someone is in the ICU because they've received a surgery, obviously they need to have antibiotics. The importance of this lies in understanding that every remedy is a double-edged sword. There is no medicine and there is no remedy that doesn't have negative side effects. And so when you are thinking about how to treat something for a patient, it's important to moderate every therapy because every therapy, you want to moderate the side effects of the therapy and the risks of the therapy with the risks of the thing that you're treating. And so when you have something like an antibiotic where you think there's never anything bad that it can do, that is never going to be true. We might not know the bad yet, but there is always going to be a point at which you can go too far with any therapy. And so we want to think carefully about how to moderate our therapies for every patient. You want to have interventions that save people's lives, but you also want to understand that every intervention comes with its own risks. So you don't want to over-intervene.